seminar of this series for 2021. So I would like to begin by acknowledging the traditional custodians of the land I'm on today, which are the Anaiwan people. I would also like to acknowledge the Wadi Wadi people of the Darawal Nation, who are the traditional custodians and owners of the land upon which the main campus of the University of Wollongong is located, which is where I work. Sovereignty has never been ceded. It always was and always will be Aboriginal land. I pay my respects to elders, past, present, and emerging, and extend my respect to the traditional custodians of the lands from which you're all joining me from today. So my name is Maddie. I'm a PhD student at the Wollongong node of the centre, and I'll be acting as chair for today's seminar. So some housekeeping before we begin. I would like to remind everyone that this seminar is recorded. And I would also like to ask that everyone please mute themselves for the duration of the seminar and leave all questions until the end to minimize interruptions. Feel free to type any questions that you have as you think of them into the Zoom chat during the seminar, and we'll read them out at the end, or you can use the raise hand function at the end of the seminar. So I would like to extend a warm welcome to our speaker, Dr. Doreen Matthies. She's joining us from the National Institutes of Health in Maryland, USA. Dr. Matthies is an Earl Stadman tenure track investigator and the head of the structural biology unit in the basic and translational biophysics division at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. Dr. Matthies completed both her undergraduate and postgraduate degrees at the University of Frankfurt, Germany, and began her career at the National Cancer Institute before joining the Cryo-EM department at the Howard Hughes Medical Institute. Dr. Matthies established her own lab within the NICHD in 2020, and her current research focuses on the structure and function of membrane protein complexes in their native lipid environment. Today, she'll be presenting on the cryo-EM of membrane proteins, what have we learned in the last decades, and what are the current challenges. So I'll pass it off to you, Dr. Matthies. Thank you.
Thank you so much, Maddie, for the kind introduction, and also the seminar committee of the Center for Cryo-Electron Microscopy of Membrane Proteins for inviting me to the seminar series. So thank you. I've been working on membrane proteins for, I think now over 15 years, and I'm hoping with today's seminar that I can share some of my experiences with you and what I've learned throughout this time, and more importantly, what kind of tools are available to you when you're studying a membrane protein complex by cryo-EM. And for those of you that don't know me yet, Maddie gave us a good introduction already, and I'm trying to change my slide. Here we go. I thought I'd tell you a little bit about my scientific journey, so you know my background and where I'm from and what I'm trained in. So as Maddie already mentioned, I studied biology at the University of Frankfurt with a focus on molecular biology, and my majors in biochemistry, genetics, and microbiology, and decided to do my one-year diploma thesis, which is still happening in Germany, at the Max Planck Institute of Biophysics, where I got introduced to structural biology and got fascinated with membrane proteins. And I was so blown away by all that I even stayed there to do my PhD. I was kind of close to Australia for a little while. I went down to New Zealand to visit my collaborator at the Otago University, which was a fantastic experience. So I know it's very difficult times right now, but I recommend to all students or postdocs, if you have labs you work with, plan a visit at least. And after it's more safe to do so, get to know other lab cultures, see the world, and try to travel a little bit. When I was finishing my PhD in 2012, roughly, I saw the huge potential in using cryo-EM as a high resolution method and apply it to membrane proteins, which I did as a postdoc at the NIH at the National Cancer Institute in Sriram Subramaniam's group. And then, as Maddie mentioned, I went to the Howard Hughes Medical Institute at Janelia campus for 2 years before actually starting my group. At the end of last year, difficult times, but getting everything set up, people have joined me, but there's still room for people to join my lab, which I'll announce later. So what is so special and challenging about membrane proteins? As many of you know, about 1/3 of all human proteins are membrane proteins, and over half of them are drug targets. So there's a lot of interest in characterizing them and solving their structures for drug development to fight human diseases. But it turns out that membrane proteins are much more challenging to study compared to soluble proteins. And here on the left, you can see a statistic of PDB entries of membrane proteins in orange down here. And you can see how compared to all proteins in blue, which is very clear and shows how much more difficult it was and actually still is to get high resolution membrane protein structures. So why is that? If you look down here, membrane proteins are embedded in a lipid bilayer, and until very recently, limiting factor in membrane protein structural biology in our field is the ability to extract or solubilize them in a stable and functional form. They have a tendency to aggregate when extracted from their lipid bilayer environment, and for many for many years to go, what we typically do to extract membrane proteins is using detergents. But throughout my talk, you will actually see that nowadays there are other ways that I encourage everyone to explore, especially for the more fragile membrane proteins and membrane protein complexes, which just are not surviving these detergent extraction steps. So what kind of membrane proteins have I looked at so far? I have a little overview here during my diploma thesis and PhD time. You can see that I looked at ATP synthesis, focusing a little bit on the membrane domain down here, the so-called rotary C-ring, where we used a variety of methods like AFM here on the left, 2D crystallization, some low resolution single particle EM work already. This is negative stain work to study the oligomerization, the oligomeric form of these rotary C-rings. And for high resolution work, we actually used X-ray crystallography to look at high resolution structures. But as I already mentioned, in about 2012, I saw this huge potential of cryo-EM with the development of direct electron detectors, energy filters, much more stable microscopes, and the development of imaging processing software. So I joined a cryo-EM lab, and many other groups as well, including ours, were able to use and test this new technology and solve high resolution structures of initially stable soluble proteins. In our case, we picked beta-gal here on the left. And then I immediately switched over to membrane proteins, where I'm very interested in, as well as smaller and smaller proteins, which we didn't think was possible to look at back then. So the first membrane protein complex I looked at was the magnesium channel CorA, which is down here. Back then still in detergent, in a detergent micelle. Later a voltage gated potassium channel in lipid nanodiscs, and you'll hear about all these examples in my talk and what I learned from them. But just to remind you how different electron microscopy was back in the days, and how lucky you actually are to be able to use today's technologies, I thought to include a slide on how I used EM as a student and how I used it and how I use it now. So in the early days, we actually used physical film, and this is an example of how that physical film looked. We had a box at on the microscope that had space for, I think, around 50 films, and then you had to you had to work in a dark room, of course, to handle and develop these films. Then you had to dry them, you had to develop them, then scan them to high resolution, which took forever. Manually select particles back then, and only have access to kind of very basic software, like I remember iMagic. So it took me around 7 days total to collect around 100 images. I mean, the collection wasn't done in 1 or 2 days, but then it took another 5 days to just, you know, manually get to 6,000 particles that you could then play with. Thanks to, you know, what I said, the development of more stable microscopes, direct electron detectors, better software, faster computers, the energy filter and better grids lead to high resolution. That whole time reduced significantly, where nowadays on a routine basis, within a day, you probably have around 3,000 images, or now we call them movies, that depending on how many particles per movie you have, you can get a ton of particles that then, thanks to many of these factors, get you to medium to high resolution in the best case scenario. So the main message to students, whenever you get a little impatient because an experiment takes long in your view, especially for EM, but I guess this is true for many other fields as well. Just look back on what students just a few years back had to go through and how long it took them to perform a similar experiment. So I will always remember my struggles and be extremely thankful on how EM improved, and for parts that are still tedious or take longer than I would like, actually, there's room for optimization and improvement, and that's something I can work with, or the whole field can work with. So here's a graph of what kind of amazing progress the cryo-EM field has made in terms of being able to solve more and more high resolution structures of smaller and smaller complexes, with a special focus on membrane proteins that I'll add throughout my talk. So I divided single particle cryo-EM structures of 4 to 10 angstroms in blue, and better than 4 angstroms in red. And you can see that before we had access to direct electron detectors, which happened around 2011, only large symmetric specimens like these viruses could be solved to high resolution. But the development of direct electron detectors, it was really possible to look at smaller, less symmetric protein complexes. And today I'll focus on membrane proteins. And some of you may still remember some of the first structures of the trip channel and the gamma secretase in amphipols, followed by other membrane proteins like my magnesium channel and detergent. So back then, using detergent was the common method to isolate membrane proteins from lipid membranes, and the processes is illustrated in my next slide. There's a variety of detergents out there, but even the mildest detergents, in my view, tend to decrease protein stability while increasing protein aggregation. And in this representation on the left, you can kind of see why. So initially, the membrane proteins are nicely embedded in a lipid bilayer, surrounded by lipids or other membrane proteins, which is not indicated here. But once detergents are added, they start to delipidate the protein, and in many cases, dissociate protein complexes. And the end products are in detergent solubilized membrane proteins, the detergent micelles, lipid Bicels, but this is basically your product. So I hope you can see how that may not be the optimal method, but it has been widely used in the past and still is used most frequently. So the proteins that survive that step can be looked at that way. So overall, to keep in mind is that detergents can affect the functional and structural properties of membrane proteins, that detergents can dissociate protein complexes. So if you're working with a complex, you're looking at regulatory components, other ligands, then that may not be the best choice, or you have to do a lot of screening to find the one that works best for you. Detergent concentration often increases during protein concentration steps. And that's something I've seen over and over and over again, and many people still don't know about it, that actually happens. So high detergent concentration also leads to empty detergent micelles, which you see here, which increases the background noise in EM images. And while mentioning detergent micelles, I wanted to also use this opportunity to show you this wonderful paper from the Arne Möller group on detergent micelles, since this is really the most common issue during the first screening steps, if you're still using detergents, are these empty detergent micelles. They can look similar to what you would expect your solubilized protein may look like, especially if you work with a small one. And that is always why I ask people to include a control with just their buffer with detergent, concentrated the same amount that you would do with your protein, and look at it. So in this paper, they looked at the variety of detergents at different CMCs. And I think in my next slide, I have a zoom in for a very common detergent DDM at different CMCs. And even if you use 100 KD filter to concentrate your protein, this detergent will still accumulate and increase, and you will have that in the background of your sample if you concentrate or if you're not careful with your detergent concentration. But well, initially, I also use detergents. And we will get to all the methods that don't use detergents anymore. So let me come to my first example, the 200 kilodalton magnesium channel CorA, which I started to work on in 2014, and what I learned while working on it. So we knew quite a lot about this channel, and I'm trying to make it as short as I can because I want to get to these other parts. We had functional data, which indicated that the channel opens up at low internal magnesium concentrations and closes once you reach a normal magnesium concentration. There were even X-ray structures available in the closed state, which showed that the channel is a homopentamer, and at normal to high magnesium concentrations, you would expect, they even saw magnesium ions bound between these protomers. So we had a lot of knowledge about it, but what was not known is how this channel looks in the open state. And there were a couple models out there, or predictions, let's put it this way, but they all looked a little bit different. So on the upper left, based on a soluble domain X-ray structure, the model suggested that these stalk helices kind of open up a little bit. On the right here, you see a model where the open state suggests that the stalks come kind of closer together, which was based on EPR data. And then we have a structure in magnesium-free conditions where you would expect an open state that when you overlay it to all these closed structures, the blue one is actually the one in magnesium-free conditions, it looks very similar to that kind of five-fold symmetric closed state. So we thought, this is a challenging system for, it's small, it's a membrane protein, and we have, questions to answer. And hopefully we can see which of these models would be true. So I was extremely fortunate that the protein purification was already established and that this channel was stable in a variety of detergents listed here. It looked super pure on gels. So SDS gels is always a good way to check the purity of your sample. I still love to run native gels that give you some sort of idea of the oligomeric state, and if you have some sort of higher clusters or higher oligomers in your sample. So again, fantastic starting material, and I think many people still forget how long it takes to just establish a purification of a membrane protein. And for single particle cryo-EM nowadays, I would say this is really the absolute main limiting step to get your protein in that kind of shape. A quick overview of how I used to prepare cryo-EM grids, screen, and when I decide to collect data, and how I roughly process the data based on a soluble protein, the beta-gal. So you start with your very precious sample that probably took you forever to make. And back then, we traditionally put 1 to 3 microliters on a so-called EM grid. Nowadays, there are more automated systems that also spray or print. Here in this case, you still need to blot and freeze. And back then I already had access to something like the Leica or Vitrobot. And then you transfer your grids in an electron microscope and you actually look at it first. And many times after you look at it, you have to go back and repeat that step, either the grid making conditions or the sample preparation itself, unfortunately sometimes. And only once you get to images with a nice distribution of particles that you can recognize as yours, only then I would go ahead and collect data, pick up particles, run 2D classification. Back then I used to make my initial models from 2D classes using EMAN. That has changed a little bit to now, and then I process data. Back then also I used already Reliant. So this is how CorA initially looked in different detergents. Here, 3 of them, as you can see, Salmo-6, UDM, and DDM. And I'm not sure how well you can see, but if you zoom in, I talked about empty detergent micelles before. And what I recommend people to do is actually have a rough idea of what's the size of your particle and just measure. And in my case, when I looked at the Salmo-6 sample, these little particles that I saw, they were just way too small for my membrane protein. So I'm assuming that many of these particles that I'm seeing are actually empty detergent micelles. I don't know for sure, but that's my guess. And the only sample I could really identify sometimes some protein molecules that I can recognize as a membrane protein and having the shape of this magnesium channel was in DM. So this sample was still not perfect. I had to deal with thick ice aggregation and clusters, as you can see, sometimes no particles in ice, low amounts in thicker ice, low signal to noise, and probably still high detergent concentration, and the sample was just not homogeneous enough. So even at these conditions, I spent another 3 months to optimize the sample. And here I put a couple of challenges of, some of them I've already mentioned, with ice thickness. For high resolution, you really don't want to collect in thick ice. If your squares look small like this and the holes look almost darker than your carbon areas, it's probably good for an initial check and some low resolution work, but I would probably try to optimize it and try to bring more particles into thinner ice. Aggregation and clusters, preferred orientation, we have a lot. Not seeing any particles in ice. We've seen disassembly of protein complexes, low signal to noise ratio I told you already about, and heterogeneity. And these are a couple of optimization steps you can try. We have various different grid types. We can pre-treat the grid differently. We can play with sample volume. We can play with the sample buffer. You can add something just before you make grids, or buffer exchange, or do whatever you like, and just try. The protein concentration made a huge difference in the case of CorA. Temperature, humidity, filter paper, blotting time, blotting position, blotting. I have pre-plotting time and blotting time, of course, it's a separate post-plotting time as well. The plunging device I told you about, if the manual plunger or Vitrobot Leica doesn't work, we now have access to plungers that use a spray technique or printing technique. So try it out and see if that may be the magic trick to get your proteins into these holes. And of course, grid handling is a big one. So this is how the magnesium channel images looked after about 3 months optimization. And as I said, some of these things like grid pre-treatment, plasma cleaning, glow discharging, the time of it, The buffer was important, the detergent concentration was important to reduce that, and the protein concentration as well to bring that up a little bit in detergent. I will quickly run through these. Back then I used EMAN for 2D classification. Now we have a lot of software packages available to all of us that you can use, and you can play with them and see which one work best for you. But for CorA, you can see what I'm always looking for. If you're working with a membrane protein, if it's a nanodisc or a detergent sample, you should always see this kind of membrane domain, hopefully with some density inside that should be the density for your transmembrane helices. You want to see side views. Side views have these kind of rod-shaped membrane domains, tilted views, top views, and in this case for the closed state with magnesium present, I could already tell from 2D classes that I have some sort of 5-fold symmetric state. Make an initial model. And back then I was able to quickly get to a kind of high resolution where even at 3.8 angstroms as a crystallographer, it sounds low, but I got to know the resolutions in cryo-EM, and 3.8 below 4. I love these kind of resolution ranges because you start seeing the bumps for side chains, and you can even see positively charged larger ions like magnesium at this stage. But this is not the point to go into structures. We wanted to know what's going on with that open state, and just looking and comparing these 2D classes already gave us some indications. We had some side views that looked different. The soluble domain looked, had almost like 3 domains, so looked wider, but some of them looked like they are more narrow. And what turned out to be is that I expected a symmetric state because it's a homopentamer, but relaxing symmetry actually led to a very large asymmetric state. And initially having maps like that, and when I started, putting, modeling in a structure, I noticed additional densities, including helical looking densities here, which indicated that I had an ensemble of structures merged into one map. So I knew that I had to classify further to try to separate these different conformations. And I had to, stop after getting around these 2 classes, which got me to the end of the road with less than 30,000 particles from my data set back then. I'm pretty sure there were other conformations in there, but all of them were asymmetric. So the take-home message here is, if you're working on a homo oligomer where you expect symmetry, try to still relax the symmetry and see what you get. Another more recent example of a membrane protein complex in detergent, I think here we use GDN, where releasing the symmetry led to many more insights than with using symmetry during processing. Was this beautiful collaboration with Biao and Olga Budger's lab at Cornell. When Biao processed everything using threefold symmetry, he obtained an average structure that showed 3 inward-facing protomers of this amino acid transporter. But when he used the new reliant features of symmetry expansion and signal subtraction and classification of these individual monomers of this homotrimeric transporter, he discovered individual protomers in the outward-facing substrate-bound conformation. So the take-home message again is really, in case you're working on a symmetric system or homo oligomer, release the symmetry and check for slightly different conformations, and you might find something really interesting. But I told you detergents may not be suitable for all membrane proteins. Only the most stable survive, and there's some evidence that amphipoles and detergents can restrict the conformational flexibility of membrane proteins. So what other tools do we actually have available? In the meantime, actually quite a lot, and I tried to add most of them in my next slide. So most cryo-EM structures, as I said, of membrane proteins so far have been solved in this kind of detergent micelle or amphipoles. But recently, I'm super happy to see a shift in more and more structures coming out that are in some sort of lipid environment. If you know what lipids are needed or bound to your target protein, you can simply add some of these, which may stabilize your protein a little bit. We call that a bicell. But the most common method these days is reconstituting your protein into lipid nanodiscs using these MSPs or membrane scaffold proteins, which are apolipoprotein based, and a whole library of different constructs of different lengths and properties forming different sizes of discs is now available thanks to the efforts of the Sligar and the Wagner and other groups. So let me give you an overview of how this method is used. It unfortunately still requires the membrane protein to initially be solubilized in detergents, but then with the addition of solubilized lipids and the membrane scaffold protein and the removal of the detergent using either biobeads or dialysis or other methods, you can form these synthetic, I call them synthetic nanodiscs because I want to make a difference or a distinction between synthetic and native nanodiscs, which I come to later. So where your membrane protein is basically surrounded by lipids of your choice. In about, I think it's 2006, 16, I looked at this voltage-gated potassium channel in lipid nanodiscs, and it wasn't really because I wanted to step away from detergents back then. I didn't think about it because I was a crystallographer. The reason for it back then was to look at the interaction of the channel activating toxins in complex with the channel, which they had found to interact with the lipid bilayer. So we really, really needed this lipid environment and chose a slightly larger disc so there's enough area of lipid around so that toxin can basically bind properly and have that lipid environment. And Sheng Yung did a fabulous job doing all these initial tests, which you have to do when you want to reconstitute a membrane protein into nanodiscs. You're going to have to find the kind of perfect lipid to protein ratios. I see a lot of people just adding a ton of probably too much lipid, and they elute most of their, proteins in something close to the void. So I recommend you initially can calculate your, how much lipids should be in there, how much space would your protein take, and take that out, but still do a plus minus ratio, do some optimization, and he really did a fabulous job optimizing this for this KV channel. Then, one important thing I would also do right after the protein purification is negative staining. No matter what lipid disc technology you're using, just to check how homogeneous that sample is. You can also do that with detergents. You can really learn so much from your sample if you do this half-an-hour experiment. It's extremely, not very time consuming. So on the left, and so I did that exactly for this KV channel. On the left, you can see such a negative staining EM image on this potassium channel reconstituted in lipid nanodiscs. And the beautiful thing is that you can easily identify the lipid nanodiscs already. So again, little rod shapes here. And again, you can do this within half an hour, quick negative staining, letting your grid dry, and then putting it in a basic microscope like a T12 or T20 we have available here. And if you have a little bit more time, you can collect a couple tens or 100 images, picking up your particles and classifying them in 2D, which I did here. And in this case, we observed a subpopulation actually lacking the soluble beta subunit. I've skipped that part. It's this KV channel has a membrane alpha subunit with a little bit of a soluble domain and then a soluble beta subunit. We couldn't really see that really well in the gels, but I can clearly see it in negative stain. So I knew my population isn't homogeneous. I also saw that the soluble domain compared to the membrane, that rod shape, the membrane domain, is kind of tilted a little bit in some of the particles. And what you oftentimes see in nanodiscs is that your particle doesn't sit in the perfect center of your disk, but it actually has some room to be placed in different areas of the disk. So it's not perfectly centered, which you don't have to worry about once you get to high resolution. But these are good things to observe and know about, especially this kind of tilting business, because that helped me a lot using, thinking about a strategy of how to process my cryo-EM data to high resolution. So we did not get the cryogrid preparation right away. We did notice a lot of soluble contaminants in the sample, and in the ice, and the membrane proteins kind of sticking to carbon regions, which is another thing that can happen, and that is a challenge when you're using these standard quantifoil grids. So here we switched to ultra AU foil gold grids, which shifted kind of the ratio of membrane proteins in the ice versus probably somewhere bound to the gold, I don't know, to a higher ratio. So in these cases, I saw definitely more membrane proteins. And what my take-home message here is, you may have a pure-looking membrane protein based on your SDS gel, but also make sure you can see typical membrane protein features if you're working with a detergent micelle or with these lipid nanodiscs. They are so easy to see, especially the side views. You should always see these kind of rod features. So if you don't see them, and I've worked with a lot of people bringing me membrane protein samples or bringing their grids, and they weren't really sure about the size, and they haven't really seen these features, and then they're very disappointed if they don't get a structure. So look for these features. Membrane proteins are super easy to see. They're even better identifiable than soluble proteins in certain views. So make use of it and use that. So I told you about the tilting of the soluble domain to the membrane domain. So initially, when I tried to solve the 3D structure of this complex, I ended up with some sort of structure with lots of low resolution regions. But because I knew about this tilting business of the soluble domain, I made use of some of these new features in Relion by using basically adjusting my data processing strategies in a way to improve the local resolution by extensive classification, masking, focused refinement, and using signal subtraction. So here, in this case, I subtracted the membrane domain and ended up with a much higher resolution structure of the soluble domain. Here I did the reverse, subtracted the soluble domain, and could definitely improve the resolution of the membrane domain. So make use of these features, check your negative stain, or get some low resolution data, and then think about what can you do to improve. And I really like the local resolution filtering that many programs and software packages now included. That will also help pick up the higher resolution regions and filter it to higher resolutions if you actually have it. But this was, of course, not the first membrane protein structure in a synthetic lipid nanodiscs. The most well-known and the one of the first high-resolution membrane protein structures in lipid nanodisc was the trip channel, resolving some tightly bound lipids, which was super exciting. But looking at structures in nanodiscs, there were actually a couple already published before, of course, at lower resolution. For example, that translocon, the integrin. And I really like to show this example of this membrane V-ATPase subcomplex, which went from low resolution using kind of older technology to super high resolution, thanks to basically the new technological improvements in the field of cryo-EM. And here I have a couple more examples of different structures solved in lipid nanodiscs, and I apologize if I have not selected your favorite one, but this method works. If you want to add back some lipids, this is a wonderful method to use, and I always recommend it. So these were examples of nanodisc structures using these traditional membrane scaffold proteins. But there are similar methods out there that do not use these MSPs, but subposin lipoprotein, or here on the right, these peptide discs, peptides. The procedure is very similar to the nanodisc reconstitution process. And here I have a few examples of these systems. So first, I think I'm showing the Salipro examples, and later on some of these peptide discs. So MSP, or membrane scaffold protein, just to remind you again, is based on apolipoprotein. Salipro stands for subposin lipoprotein. So subposins are known to be modulators of lipid membranes, mostly at an acidic pH within lysosomes, and with their lipid binding properties, they can be used to form these lipid nanoparticles. And especially in the last few years, some labs have successfully used these subposin lipoproteins. And in the case of this calcium uniporter, which I oftentimes like to show, you really are seeing how 6 of these subposin molecules wrap around the lipid membrane part of the protein. I'm also highlighting a recent example from Misha's group on the various conformations of a serotonin receptor, which are known to be modulated by the lipid environment. So it's important to have some lipids there, I think. So compared to previously published conformations in Detergent, the lipid bilayer stabilized this receptor in a more tightly packed coupled state. And besides finding in asymmetric state, which suggests in asymmetric activation of this receptor, they could also resolve the agonist bound receptor conformation, which adopts a wide open pore capable of conducting sodium ions. So, so one thing I've heard, so this is fantastic. So this is great, and it seems to be, you know, a very similar technique to these MSPs. One thing I've heard about the system, and I haven't used it myself yet, is that there seems to be some certain degree of lipid preference for subposin A at physiological pH. So that's something to be aware of when testing that system if you're using some special type of lipids. But I would still give it a go. This looks, many of these structures look incredible, and probably more native than in detergents. Another similar approach are these peptide discs, which are again based on apolipoprotein, similar to these MSPs, but have a reverse sequence and other residue replacements to increase lipid affinity. And with its, you know, much smaller size, multiple copies basically wrap around the lipid membrane part, which you can see here in the 2 examples of MSB A and MSCS. These are 2 examples from the Tom Vals's group. But as I said earlier, all these 3 disc-like approaches still require the initial solubilization using detergents. That is why I'm super excited about the so-called native nanodiscs, which were developed already about a decade ago. I only learned about them a few years ago. And now we have a variety of these amphiphilic copolymers available to synthesize ourselves or purchase from, you know, purchase them commercially. And the main challenges I'm hearing from students is still that they may not solubilize your membrane proteins as well as detergents. They seem to form heterogeneous sized particles, and that it's just not very easy to work with these polymers. For example, on a size exclusion chromatography, it seems to be a little sticky. But luckily, there are many different groups that work on new classes of these polymers, including these cyclopoles, which you can find in this reference down here. We're also currently in the process of testing all these different polymers, and we hope to find something that works for most of our bacterial as well as mammalian systems. And together as a whole field, the more data we have, the more we share our experiences and then what we find, the better it will be for, for future generations. So these polymers can be used to solve high resolution structures. It has been shown. And my next slide, I'm going to show you how these polymers work and some of these examples of structures. So on the top, I'm showing you roughly how these copolymers spontaneously insert into membranes and break them up into these lipid nanodiscs, which can then be further purified. Here, the beauty is that no solubilization step by detergents is necessary, and using these polymers basically leaves a patch of native lipids, lipid membranes around that hopefully never destabilizes them. So these are the examples from bacteria here with ACRB and the supercomplex of complex 3 and the cytochrome C oxidase. But there are also examples from mammalian membrane proteins here. I'm super happy to see 2 examples from the Eric Gore lab with these 2 examples here on the right, expressed in hex cells. I think those membrane proteins were incubated with 2% SMA, so that the styrene malic acid polymer from Polyscope, showing this chicken acid sensing channel and the glycine receptor and a variety of conformations, just published this year. Another system available for structural biology of membrane proteins are liposomes, where you reconstitute membrane proteins into lipid vesicles. It's a very well-known method for using it for functional studies, but we can also use that for structural studies. But as I said earlier, all of these disc-like approaches, and as well as unfortunately the liposome system, the membrane protein has to be solubilized before. But I wanted to show you a few examples here. Again, I talk a lot about Misha's examples, but Misha looked already at the serotonin receptor in tightly reconstituted proteoliposomes by cryo-electron tomography and subtomogram averaging, getting, I think in this case, it was like a 12 angstrom map. But below here, there are more examples. And one of the first ones I think I've heard about was that that was a, it was a channel. I think it was also, it was a potassium channel in liposomes in the lab of Fred Seekworth at Yale that I don't show the structure here yet, but they pushed it to higher resolution as well. I added this example here of this macrophage expressed gene 1, which is not a typical transmembrane protein, as you can probably see, but it can form pores in target membranes like bacteria upon acidification. So in the presence of these liposomes, they studied its interaction with the membrane and could solve one of these states to 3.6 angstroms bound to these lipid membranes, which I found super exciting. So they use liposomes here. Then we have an example of a ATP synthase, which is an ATP synthase former researcher I enjoyed seeing. But the ultimate highlight of the structures in liposomes was, of course, ACRB, again, which could be solved to 3.9 angstrom resolution. So definitely also a method to look into. The only thing to be aware of here is that in order to produce really thin ice, you would typically aim to make small liposomes. And you can see here they're rather on the small side, which then leads to a large curvature of these membranes. So that still has to be, seen how it affects the structure and function of some of your target proteins. But another method that you can try to use. So summarizing the progress the field has made over the last few years, here again, a graph with the selection of membrane proteins in different systems, from detergents to amphipol to these MSP nanodiscs, Salipro, Peptidy. So far, there are only examples of this polymer SMA or the liposome example, I think, is here with ACRB. But these are only a few examples. The group of Manuela Soames in Paris actually looked at a lot of published data of membrane proteins and classified them into different groups. And I recommend to look at her review if you're interested in seeing all these details. She really split it up in different detergents, the MSP base, the Peptidy discs, and so on. And you can see the different amounts of structures. And I think her limit was 5 angstroms, but check out her review. A lot of work, a lot of analysis went in there. And this is something to check out. And she's also the group that works on these cyclopoles, so a new class of polymers that may work even better than these SMAs. I still have a few minutes left, so I want to mention the great potential of looking at much more crowded subcellular fractions or cells themselves, and mention a couple of examples. On the left here is isolated rough ER fragments. And basically, they were able to use cryo-electron tomography and subtomogram averaging and classification to get quite good resolution information on these ribosome docking sites, for example. Still, I'm hoping that this method will be able to be used to push the resolution a little higher. But this is another great option to look into, just to use native subcellular membrane fractions of your choice. Here on the right, where they looked at thin areas of even intact cells, intact small cells, and using template-free classification to pull out maps of different membrane protein complexes in such a crowded environment. Definitely a method to watch, follow, and try yourself if you can. For samples that are too thick to image, they can be thinned down now. A method that has been established, we call these cryolamella, that we then add in the electron microscope, thinned down samples from thick samples. And then you can perform either the single particle approach or tomography and use subtomogram averaging. And here I'm showing the two best known examples, at least, of membrane proteins so far. You've all heard about the ribosome and proteasome and things like that, but looking at membrane proteins, these are of course slightly lower resolution due to various challenges, such as lower numbers of particles, crowded environment, and a low signal to noise ratio, but it shows it can be done, even though these are examples of very well recognizable cellular components, like the nuclear pore or the translocon complex. But it can be done, and one day we will maybe only be using this in situ structural biology technique. But the challenge is, of course, to find your target of interest, and many other challenges I think that I have summarized in this kind of boring slide. And I've mentioned a lot of challenges or things to look out for on the way. But I think I just made that slide 5 minutes ago. It's not really pretty, so I'm just gonna try to summarize an overview of challenges, but I'm hoping that you guys have written down some notes or rewatched that video to see what I've kind of mentioned for these different systems. So just be aware, if you're still using detergents or amphipols, that they can contribute to protein instability, complex dissociation, a limitation in conformational flexibility, and you're definitely getting higher background noise in cryo-EM images. So be careful with the amount and the concentration of it and that type. Synthetic MSP lipid nanodiscs still expose the membrane protein to detergents initially before you reconstitute it. And they typically show more heterogeneity in form of the nanodisc size. So the size varies sometimes. And also the protein location inside the disc varies. But I would not worry about it. It can be, these two things can be neglected at high resolution. If you have enough signal from your membrane protein, it should come out. So don't worry about it too much. Synthetic lipid nanodiscs using the Salipro or Peptidy discs seem to be a little bit more homogeneous, but still the membrane proteins are initially exposed to detergents. And about the Salipro method, I've heard a little bit about lipid preference. So just watch out for that. Native lipid nanodiscs using these polymers, I hear is still challenging. There are so many different products out there with different qualities. So talk to people who've worked with them, see which companies they ordered it from. You may get much lower yields than compared to what you're used to with something like DDM. The sample, your particles may look more heterogeneous, and it seems to be more difficult to work with, but has a lot of potential since these membrane proteins are never exposed to detergents and basically surrounded by their native lipids and interaction partners. So if you're looking for complexes that easily dissociate, I would definitely give this a go. Liposomes, again, you still expose your membrane protein initially to detergents before reconstitution, same as these synthetic nanodiscs. Small liposomes for thin ice have a high curvature, and it still remains to be seen how that influences different membrane proteins. Tightly packed liposomes lead to lower signal to noise ratio for individual particles. Be aware of that. But the example of ACRB is a great one to see that it can be done, and you can actually get to really decent resolution. Isolated native membranes are heterogeneous, and the particle selection and classification will get more challenging. But I think it's worth to investigate and try to push the resolution a little bit for that as well. And in situ structural biology, using these thin lamella, these thin cryolamella is still challenging. Just producing these lamella is not straightforward. I recently started doing that. But you still have to deal also with lower signal to noise ratio in this kind of crowded cellular environment. You have low particle numbers to work with until everything gets to another scale. And it's very challenging for small proteins to identify your small proteins. So tagging, knowing where to look for it, and large data sets are probably necessary. I'm almost coming to an end, and I don't know how much time I still have, but I wanted to put this slide in. If you are looking for a postdoc position and are interested in cryo-EM or tomography of membrane proteins in a lipid environment, feel free to reach out to me by email. My lab is located on the main NIH campus in Bethesda, Maryland, on the East Coast in the US. We have multiple screening scopes and data collection scopes available, various computing resources, and newly renovated lab and office space. And of course, a fantastic collaborative environment here at the NIH with access to a huge variety of basic and as well as clinical research, which is probably a highlight here at the NIH. And here are a couple of recent and unpublished projects. We work on lipid metabolism, various protein complexes with a focus on membrane proteins, of course. For the last 1.5 years, we've done a little bit of research on some of these SARS-CoV-2 membrane proteins, as well as the spike variants on pseudoparticles. So we're trying to do our part in helping out here. But then I think I'm running out of time. So with that, I think I would like to finish and acknowledge my growing team members who were so brave to join my lab so far during this terrible pandemic. Also the NIH Intramural Research Program, Cryo-EM Consortium for all their support. My previous labs and all their lab members, I learned so much from them. and of course all my collaborators, which are listed here on the right, and the NIH and NICHD for all their funding to establish my own lab, and you for your attention. And if there are any questions, I'm happy to answer them now or by email later. Feel free to reach out to me. My email is down here. And with that, yeah, I'm happy to take any questions.
Great, thank you so much, Doreen, for that talk. It was so interesting, and I'm sure I speak for everyone when I say that. So we have run out of time for today's seminar, but do you have time to stay back for a few minutes for some questions?
Of course, yes, of course I can stay here.
Great. So before we get into that, for everyone who does have to leave us and doesn't have time to stay back for the questions, I hope you all have a happy holidays and a safe time, and I hope you all are relaxed, and I wish you all the best for next year. Okay, so some questions we had. I think the first one we had was from Rajiv. So Rajiv, if you wanted to unmute and ask your question, that would be great.
Hi, Doreen, great talk.
Oh, thank you.
Yeah, I was wondering, how did you find back then the in symbol of a structure, and how did you classify at that very time?
Sorry, say that one more time.
I'm wondering about the in symbol of a structure on your map. How did you find back then?
Oh, the ensemble. Yes. Okay, I can explain that. Let me go back to the slide. You mean this one, right?
Yeah.
Yeah. So I basically started putting in protomers in segments, and filled my helices based on the closed state structure. And then I basically colored everything that was surrounded by protein in one color and in a certain distance. And everything else that was too far away from my model, from the PDB, that's kind of not visible here, it's inside the density, I colored in a different color. So you can see, for example, extra density that is clearly from the detergent micelle. But I had these two rod-shaped densities, which looked like helices to me. I was like, do I have any peptides or helices bound to it? Like initially I was totally confused, but then thinking about it made me go like, wait a second, what if this subunit has different conformations? What if the subunit actually can be more inward, basically leading to these helices being pushed further to the inside? And that made me think, well, I may have multiple conformations in there that I can probably separate, because these differences should be able to, I mean, I was hoping that the software can identify these differences. And I was so lucky that this actually happened by then. And you can now see here, this was kind of the merged structure initially, but I pulled out this conformation, which had kind of an inward movement, and these helices basically much closer to that subunit B, for example. So the location of subunit C and D compared to, you know, B here, you can see that it's very different. So it was kind of seeing extra density after fitting in a structure that density that was not accounted for in my model, and seeing that it's helical, and putting two dots together and trying things.
Great story. Thank you.
Okay, great. Thank you for that. Our next question was from Anisha. Would you like to unmute and ask your question?
Yeah, hi. Thanks, Doreen. Great talk, and thanks for sharing all your insights. My question is, what have you found useful in terms of dealing with dissociation of complexes, and not necessarily these are with detergents? So even without detergents, if the complexes are dissociating, if you have any insights, that would be great.
Yeah. I wonder if I should, I mean, still, like in the past, we still used a lot of detergents, and if I knew something was a complex, let me go back to CorA, because that was actually interesting. So if you know you have a complex, it's useful to, of course, run gel filtration profiles and kind of estimate your size, but also these native pages do give you a good overview of what your size is. And if you use a detergent that is not really good. You can even do a time-dependent native page, over maybe multiple days. And what we actually saw for CorA, that if we had CorA and amphipol, we actually dissociated CorA into its monomers over time. So we were hoping amphipol will be the magic bullet because of the trip channel and the gamma secretase structures, and we put it in amphipol as well, but we weren't as lucky. And we saw that through, running these native pages over time and seeing a dissociation. And for detergents, yeah, it's screening. On SDS gel, you will always see your monomer, but native pages do give you that additional information about your oligomeric state. For an example,
can I just quickly, I wanted to know like more in terms of freezing. So if you see dissociation.
Oh, I see, freezing. Yeah.
Yeah.
Oh, freezing. Oh, good that you should have stopped me earlier. For freezing, yeah, we do see a disassembly of proteins. They look perfect before on gel filtration and native pages, and some proteins just it's just a little rough using blotting or using the forces that are going on during this plunge freezing. If it's using filter paper or others, it's just probably a very very forceful way of to these proteins. There's a lot of movement going on. You have interactions with the air-water interface that may lead to dissociation. So again, you can try to add things, different types of detergents at much lower than 1 times CMC, to see if you can kind of minimize this kind of interaction of proteins with the air-water interface. You can try the different plungers that are out there with spraying, where you basically avoid the blotting with filter paper, or the printing technique, and see if that's something your protein survives. Yeah, it's playing with buffer conditions. I have not solved a structure yet with Vitrojet or chameleon produced proteins. I've kind of managed to find conditions that work for me using the traditional filter papers, and I've played with adding additives to my samples.
Great, thank you.
Thanks for the question.
Maybe one last thing I can mention is the proteins that are really falling apart when you look at them. You can use cross-linking techniques as well. A great method to look into is graphics, where you mildly cross-link something and hope that it stays together. It's something I would try to avoid, but if you can't, then look into different cross-linkers, or glutaraldehyde as a quick and easy check. That's something I could add.
Great, thank you. Okay, so I think we'll go to someone who has raised their hand. So Isabel, do you want to ask your question?
Sure. Hi, Doreen. Nice to see you again. Thank you for this beautiful talk. I guess my question was whether you could give us any insight on expression system for membrane protein between bacteria, yeast, insect cell, baculovirus, mammalian cells, or what's best for yield and preserving the biological relevant active state of a protein.
Yeah, no, very, very good question. So I decided in my lab to still have the E. coli expression system up and running, and that's something I usually check for membrane proteins of at least bacterial nature, because you tend to be getting a lot of protein, and also your MSPs or any other proteins, you usually produce them in E. coli to quite high yield. We do have Pichia expression as well. I decided not to establish insect cell expression and went straight to hex cells for mammalian cells. So the yield, I know, it can be low, but there are many methods, and you can try to check the paper on the glutamate transporter. There, they also used hex cells and found a good way to overexpress it in a quite high yield. And that system for mammalian proteins, I would definitely recommend. Just screen for high yield and try to look through the literature of what kind of tricks and stuff you add to the medium that then leads to high expression of your protein. So that's something I do in my lab as well. I know my collaborators still work with insect cell expression and get high yields for that, but I had to make a decision and I went straight to the hex cell system. Also, yeah, that will guarantee all your post-translational modifications and so on, which you will likely not see when you use E. coli. So, yeah.
Great, thank you. All right, so next question from Gokhan, please.
Hi, Doreen. Very nice to see you again. How are you doing?
I'm good. Nice to see you. How long has it been?
4 years.
Oh my God, yeah. Time flies.
Yes, it does. So what I was wondering is you showed the particles on the liposomes, right? And you already mentioned it gets trickier because the ice is thicker and so on and so forth. So how easy is it to pick particles from those liposomes? I was wondering.
Yeah. Good question. We are in the process of, we have a few projects where we start doing it, and it is definitely more tricky. But the good thing is there is, particle picking programs are out there. You can try them, and wherever you have, you can check your density of proteins that you reconstitute into your nanodiscs. If you have a membrane protein that has a soluble domain that sticks out, that's probably perfect to see. Otherwise, you're looking for increased density and you pick it up and then you let 2D classification do its thing. So probably over picking strategy along the membrane if you're confident that you reconstituted quite tightly. I'm staying away from template-based picking just because I had a bad experience in the past, but I should still give it a chance and let students try it because the algorithms and all that got so much better. And I just don't want to see a structure of my template, which happened to me in the past. And that clearly came from noise, but I should let go of this because it was such a long time ago. So yeah, look into the ACRB paper or other papers, how they do it, and start from there. And many software packages now integrate the different picking strategies that are hopefully adapted to these systems as well.
Yeah, thanks much. And you are so lucky to be working with Rick. He's fantastic. I miss working with him. Say hi to him.
I'm very happy that I get to work with him again after working with him at Janelia. He's amazing. Yeah.
See ya.
See ya.
Okay, thanks, Gokhan. So I think the final question is from Mary Elena. Would you like to unmute?
Oh yeah, hi. So thank you for sharing all this information and all this experience you have with us. So my question was on nanodiscs. So how are the hydrophilic, the soluble domains of a transmembrane protein would be affected by the presence of lipids around it? How will this affect the outcome? And could a lipid concentration optimization rectify any issues arising from that?
Okay, I'll try to answer, but if you notice that I'm not answering your question, feel free to interrupt me again. Okay. So membrane proteins originally are basically, they are embedded in a lipid membrane. So what you're doing to form these lipid nanodiscs is basically the reverse step of removing that detergent again and replacing it with the lipids you think are important around your membrane protein. Many people, the standard techniques is actually using synthetic lipids, but you can also use native lipids that you extracted from the organism of your target protein and use these during the reconstitution process. And that should mimic the native environment, even though the belt protein around it may constrict the movement, basically the conformational flexibility of the protein inside this if the disc is too small, or even if it's larger. There have been reports that because it's kind of crammed together, that you don't have the flexibility in these discs that you would have in a native membrane. That's why going to a native, completely native system, in situ situation, or the liposome system, that may have an advantage because your protein may have more room to expand to all its conformational landscape. But it is so much better than having it in that kind of artificial detergent environment that really is very different compared to a lipid environment. I hope I answered your question, but if not, feel free to ask a follow-up.
Oh, no, you did. Thank you very much.
Okay, great. So that was the last question. Thank you again, Doreen, for your time today. That was such a great talk. And thank you everyone for staying back for the questions, and to those who asked questions, thank you. So yeah, I'll just say again, happy holidays, and